both inside the VA healthcare system and outside the VA healthcare uh, system. Diagnostic codes provide a foundation for diagnosis. For many conditions, diagnostic criteria have evolved over time, but having a first ICD code uh, based on the Kansas case definition introduced by Dr. Lee Steele provides a basis for adjusting criteria as evidence evolves. It is not at all unusual in the history of medicine for diagnostic criteria to evolve, but the Kansas criteria modified were what was presented to the CDC. That is what that is what the panel approved this code based on. We understand that VA is doing some work. I'm not sure if that's continuing or not. It's been going on for years to create a better basis of diagnosis. But until then, we now have a diagnostic code. It is based on the Kansas criteria, and should diagnostic criteria evolve with better research, then there's ample opportunity for going back to the CDC's panel and changing the diagnostic criteria as it evolves. But that is the best of what we've got right now, and that is where we are. So for those are really all health related, but I know that questions have come up as well regarding veterans' claims for VA service connection or disability compensation, as it's nicknamed. Since 1994, there's been a lot of discussion in the veterans community about all of this. Undiagnosed illness, there are two pathways to service connection under 3.317 of 38 Code of Federal Regulations for Gulf War veterans, but also now expanded under the Honoring Our Pact Act to a broader array of veterans, including those who served in Afghanistan and elsewhere. I won't go into all of that into all of that eligibility criteria. It is available on the Veterans for Common Sense website if you're interested, the VA's website, or the Veteran Service Organization, major veteran service organization of your choice. Since 1994, compensation has been available to veterans for undiagnosed illness. In 1998, fellow Gulf War veteran Paul Sullivan, myself, and a number of other Gulf War veterans were involved in helping to pass that legislation. It greatly expanded the opportunity for research, for treatment, and for benefits. In 1998, the modification then was in Section 1602 of the Persian Gulf War Veterans Act of 1998, which then became Section 1118 of 38. Of U.S. of 38 United States Code. Expanded it to an illness that is either diagnosed or undiagnosed illness. And for those of us involved with that legislation, I know for myself, I thought that was the be all end all, and that was going to be the end of it for Gulf War veterans. It took care of everything. But as we were to learn, that was really just one more step in the process. So undiagnosed illness is an availability for veterans under current VA code, but the denial rate has been extraordinarily high. 92 to 93% was what it was for many years for a denial rate, so a 7 or 8% approval rate. But there are also chronic multi-symptom illnesses. The nickname is MUCMI, M-U-C-M-I. Those are examples of those are chronic fatigue syndrome, fibromyalgia, and irritable bowel syndrome. And a natural place for the diagnosed Gulf War illness to fall is under existing law and existing code is under as a MUCMI, a medically unexplained chronic multi-symptom illness. Veterans for Common Sense would very much like to hear from any veteran who applies for Gulf War illness as a diagnosed illness. If you've been given the diagnosis, it's in your medical records, you now apply as a medically unexplained chronic multi-symptom illness. And if it is denied on that basis, we want to hear from you and we will advocate on your behalf. Those are the large impacts for veterans dealing with Gulf War illness, health related, research related, for Gulf War veterans claims moving forward. We would be open to any questions in the chat. Also available, I put our email address, info@veteransforcommonsense, in the chat. And I also posted a link where we've got a 3 or 4 page question and answer sheet about Gulf War illness that you can use for your reference. It's oriented towards clinicians, researchers, and veteran service organizations. Hopefully answers a lot of questions there. If you find others that should be answered and added to it, we are happy to consider those as well. So with that, I'm going to conclude the panel. We would again welcome any questions to any of the researchers there or myself. And thank you again to Dr. Klimas and all of your team at Nova Southeastern for hosting this conference, and particularly to Dr. Beatrice Golum for leading this effort, to Dr. Lee Steele for being just such a core part of all of this, and to all of the other researchers involved and clinicians involved in getting this diagnostic code, which is life-changing for Gulf War veterans. Thank you so much.
That's great, Anthony. Thank you so much. McKenna, I'm just going to go ahead and segue. So
Yep, absolutely.
I'll take on the next session. So the next session, I'm the chair. So it's the current research results section. It's kind of a big session. We're already running a little behind time. So I'm going to do the introductions just one at a time. Too many people to do all at once. So I'm going to start with Dr. Steele. Lee Steele, you know, not exactly an unknown person to you all, since she's been at this from the very beginning. She's a neuro and environmental epidemiologist, a professor at Baylor College of Medicine in Houston, where I did my internship, by the by. Working with collaborators across the country, her current research includes studies of deployment factors that contribute to long-term health outcomes in veterans of the Persian Gulf War, as well as neurologic, immunologic, and genetic factors associated with the Gulf War illness. She previously directed the Veterans Health Research Program at Baylor University's Institute for Biomedical Studies. She served as scientific director for the Federal Research Advisory Committee on Gulf War Illness. And she was the director of the Kansas Persian Gulf War Veterans Health Initiative that gave us the case definition that we rely so heavily on. She received her doctorate in epidemiology and human ecology at UT School of Public Health, Texas, and investigated complex chronic illness as an epidemiology fellow. That's where I first met you when you were working there with the MECFS group at the CDC. With no further ado, Dr. Steele.
Okay, let's see if we can get my slides here. Yeah. Is anyone seeing slides?
Yep, we can see them.
Okay, that's what we want. Thanks.
Wonderful.
And thank you all for that morning session. I think it gave us a lot of insights, and I also was glad to hear Dr. Golum's discussion of how veterans are experiencing their clinical treatments. And thank you to Dr. Klimas for hosting this important conference. I'm so glad that this is available every year, and it's one of a kind. So thank you for that. Today, we're going to switch gears. We're going to talk research now, and somehow we're starting with a pretty complicated kind of research question that we addressed with this study. And so it's a little hard to squeeze it into 10 minutes, but I'll hit the highlights and just let you know that if you have questions about details, please put it in the chat or contact me later and we can fill you in. So this is a study from a great collaborative group looking at exposure effects and how they differ by PON1 genetic status in relationship to Gulf War illness. So just as an overview of what we'll cover, we're going to look first at the big picture of how military exposures have been shown to be associated with Gulf War illness. But then we have to ask, why did some veterans develop Gulf War illness, but others who had those same exposures didn't get Gulf War illness? And there have been a number of studies looking at how genetic differences between veterans can help determine what the response is to toxic exposures. So for this particular study, we focused on the PON1 enzyme and genetic variants and how it interacts with exposures and results in Gulf War illness. Okay, so just have to take a minute. This is February 27th, tomorrow is the 35th anniversary of the completion of the ground war ceasefire, a great victory in the desert for our Desert Storm veterans. So thank you all. Short war, relatively speaking, and highly successful war. And yet, despite that, veterans still came home sick. So initially it was called Gulf War syndrome. This is a description from a veteran who gave testimony. His symptoms began while he was in the Gulf. Um, he had terrible headaches, abdominal cramping, diarrhea, went back to the base in Germany. It all persisted. And over time, things got worse. So he actually describes what he experienced, and that's pretty much how it looks when you look at current definitions of what we now call Gulf War illness. These are the symptom domains in the Kansas case definition, and I'm sure a lot of the veterans in the audience can relate to this profile of symptoms. We see a consistent profile across different populations of Gulf War veterans. So it's a good core group of the symptoms that they are experiencing. Now, what caused it? Well, again, a long list of possible hazardous exposures during the Gulf War have been proposed and looked at in many, many studies of Gulf War veterans. And I'm sure, again, that you're familiar with these lists: chemical weapons, depleted uranium, vaccines, pyridostigmine bromide. Well, what I can tell you from the studies, and as I said, there have been lots of studies of many veterans populations looking at the links between exposures and Gulf War illness. And there are three that consistently come up in many, many studies. And these relate to chemical weapons exposures, heavy use of pesticides and insect repellents, and also the use of pyridostigmine bromide pills, or the NAP pills. And just to give you a quick reminder of what those are, the veterans watching this can look at that picture here in the upper right. This is a picture of a veteran taking pyridostigmine bromide. These were in the foil packets, the blister packets, the little white pills. They were ordered by command to take them at certain times, and they can contain a medication that was supposed to protect against the deadly effects of nerve agents. So it's one big factor. The pesticides that veterans encountered are not your regular pesticides that you get home in the backyard. There were many, many pesticides used, and used extensively. And in fact, 64 different pesticide products have been identified by Department of Defense, 37 active ingredients, and study after study show that the use of these pesticides is linked to Gulf War illness. And then, of course, fairly unique to the Gulf War, were the chemical weapons exposures. Many reported incidents, many investigated. The most evaluated and researched one is a chemical weapons exposure following the Qamishli pit demolitions in southeast Iraq. And DoD estimates are that 100,000 troops were likely exposed to low levels of nerve agents as a result of that one incident. So these are not trivial at all. Gosh. um, So just to summarize the exposures of greatest concern here, PB, pesticides, and chemical nerve agents. These three exposures are not unrelated to one another. They're all considered potential neurotoxicants, and many of them fall into the class of compounds called acetylcholinesterase inhibitors. This is organophosphates, carbamates, and some related compounds, and they affect levels of the neurotransmitter acetylcholine in the body. Bigger topic to address, but just we'll leave it there. So that question about why did some veterans get Gulf War illness and others didn't? Well, if you're considering these exposures, there are some enzymes that help your body detoxify these exposures when you encounter them. And a major one is an enzyme called peroxinase. And one version of that is the PON1 enzyme, and there are several genetic forms of that. And for example, the Q form of the PON1 at position 192 is known to effectively neutralize the nerve agent sarin, right? But the R form of that same compound is not effective at all at neutralizing sarin. So you can imagine then why two groups of veterans encounter sarin in theater, and the ones who have one genotype may have done fine and the other genotype didn't do so well. And actually, Dr. Robert Haley in Dallas has been studying this for years. He started studying the enzyme in the late 90s and then the gene for PON1, and he has consistently found that there's a link of greater rates of Gulf War illness among veterans who carry the R genotype and were believed to be exposed to nerve agents. So his research is the first to sort of verify that there is a genetic link there. And there are also a number of other genes that have been identified by different studies, but it's more than we want to cover right now. But I'll just put them down in case you want to check them later. Um, to summarize the big picture around the PON1 gene, the PON1 at position 192 of this gene, the Q variant, is effective at breaking down nerve agents, but not effective at breaking down some pesticides. The R variant is effective at breaking down some pesticides, more effective than other forms, but it's not effective at hydrolyzing the nerve agents. So you can see sort of a dichotomy just with those two. All right, so on to our study and what we looked at. This study came out about a year ago and has just a stellar team of collaborators. The study lead investigator was Linda Chow in San Francisco. We were so fortunate to have Clem Furlong's team. He's one of the preeminent experts in the PON1 enzyme and gene. He's at the University of Washington, and his team did our PON1 work. The Boston group, led by Dr. Sullivan, was the source of so much of the blood samples, the data. It's just extraordinary. And then, of course, Dr. Klimas in South Florida, and her work with us on this was highly valuable. So let's get down to what we actually did in this study. This is kind of busy. I don't want to go through all of this, but I just want you to know that this was a multi-site study that involved 3 existing cohorts, and truly the veterans in the study came from all over the US. So the biggest cohort was the Gulf War Illness Consortium that you heard about just a little bit ago, led by Dr. Kim Sullivan, the GWIC study. This was 3 sites, her site in Boston, our site in Houston, and Dr. Klimas's site in Miami. This was over 250 veterans in this sample. And then Dr. Chow had 2 Gulf War illness cohorts from 2 San Francisco studies she had done. One was a VA supported study and one was a DoD supported study. So we ended up with 398 total Gulf War veterans. This was 295 Gulf War illness cases compared to 103 controls. We collected data on symptoms, diagnosed health conditions, and exposures during the Gulf War. And then we had the blood samples that we were able to identify PON1 status by Dr. Furlong's lab in Washington. As you can imagine, each gene location has 2 what we call alleles, because you get one from your mom and one from your dad. So there are 2 variants, the Q and the R, and then they can combine in different ways to give you 3 types of subgroups. So we have 2 Qs, 2 Rs, and 1 Q and 1 R. So Dr. Furlong identified a functional genotype for QQ, QR, and RR based on their enzyme activity in different substrates. So our analyses first looked at the big picture and looked at how all of the Gulf War exposures may relate to Gulf War illness. And then we focused down in greater detail, looking at these cholinergic exposures and how things like pesticides, PB, and being in the area where the chemical alarms are going off may be associated with Gulf War illness. Now, one challenge of understanding the effects of nerve agents is that the doses, the exposures were usually pretty low, and veterans wouldn't have known at the time if they were exposed. But if they were in areas where the chemical alarms were going off, that gives you an idea that they very well could have been exposed. So we use as a proxy for nerve agent exposure, we use whether or not you were in areas where the chemical alarms were going off. So just to give you an idea. So excuse me, let me walk you through how this worked. We looked at, as I said, we looked at the big picture of exposures. And again, like in most previous studies, the most prominent predictors of Gulf War illness were those cholinergic exposures. The PB pills, the pesticide use, and chemical alarms. So When we looked at the full sample of 398 veterans, the most prominent risk factor was the use of pesticides, and that gives you an odds ratio of 3.90. Now, for those of you who don't think about odds ratios are telling you the odds of having Gulf War illness in this case among exposed veterans compared to non-exposed veterans. Okay. Excuse me. Can't turn off my speaker. Anyway. So the second highest risk was with use of PB pills. And the third highest risk was for hearing chemical alarms. Some of the pesticides, they have different compounds, but they were not associated with the outcome. So first, after looking at all veterans combined, we focused down on the QQ subgroup of veterans. And as you see here, QQ veterans who heard chemical alarms didn't really have an elevated significant risk of Gulf War illness.
While you're coughing, remember that
sorry.
we're running out of time.
Okay, so sorry about that. Yeah. The QR veterans also did not have an increased risk of Gulf War illness with chemical weapons exposures. But the RR veterans, look at this. If you were in the RR genetic group and you were in the areas where the chemical alarms were going off, you had an 8.6 increased odds, highly significant, of having Gulf War illness. So that's how this works. We then did that with all the other cholinergic exposures. And we found that using pesticides on the skin was a significant problem for the QQ and the QR subgroups, but not for the RR subgroups. This is consistent with what we know about the biology, right? These other pesticides were not associated with Gulf War illness, no matter what the subgroup we looked at. And then the PB pills were significantly associated with all veterans combined, but then we got some kind of unexpected and odd pattern of links in the subgroup. So we'll get into that further. But to summarize, hearing chemical alarms was associated with Gulf War illness only for veterans who were RR PON1 status, and using the skin pesticides, which may be a proxy for just using a lot of pesticides for a long time, because those were the most commonly used, were the skin pesticides. But that exposure was a problem for developing Gulf War illness only in veterans who had QQ and QR PON1 status, but not people with RR, because RR is good at metabolizing the pesticides, but QQ is good at metabolizing the nerve agents. So it makes sense. Now, as I said, the use of PB pills had kind of an odd pattern, and we didn't quite understand it. Our early analyses suggested that there could be an interactive effect of PB with some of the other exposures, and this is clearly shown in animal models very frequently. So we did an exploratory analysis to try to determine is there a possible interaction here with PB and other exposures? And this is how we did it. And again, this is very simple. When looking at RR status veterans, PON1 RR veterans, those who heard chemical alarms overall had a significantly increased risk of Gulf War illness. But if you didn't take PB pills, there was no increased risk. Isn't that interesting? And if you did take PB pills, you have a substantially increased risk. So this is preliminary. This is a small study. It's exploratory only, but we use very tight and restrictive analytic methods to identify this. And it suggests anyway that veterans who are RR status, who are more susceptible to nerve agents, didn't have an increased risk for Gulf War illness if they didn't take PB, but they had a substantially increased risk, 19 times higher odds of having Gulf War illness if they did take PB. Okay. Again, pesticide use was not a problem for this group. Looking at the QQ subgroup, pesticide use was a problem for this group, but only in veterans who used PB pills, not for veterans who didn't use PB pills. So I'm going to skip the next one because there were no differences.
You are out of time. So
Oh, I am? Okay.
Get to your conclusion if you can.
Yes, okay. So I think we got the main points here. And I just want to say overall, our results really demonstrate that it's likely a complex pattern of exposures and interactions with genetic factors, prominently the PON1 192 variants, and potentially others that are driving who develop Gulf War illness during the war. Okay.
That is very interesting. And in the Q&A, if we get any time, um, there is some recent work, I think, by the Million Vet program that didn't think that PON 1 was that predictive. So I think that's very interesting that the concordant use of the prevention pills was the trigger in the epigenetic
Yikes, huh?
trigger. Anyway, we're going to move on because we need to move on. Dr. Bast, Elizabeth Bast is our next speaker, and she is an internal medicine physician at Miami VA, and she's also an affiliate assistant professor in our institute, the Institute for Neuroimmunology Medicine here at NSU, and an assistant professor at FIU. And she has a MPH from Hopkins and with a strong interest in global and environmental health. She helped to establish the new long COVID clinic at the Miami VA. And because it was using a similar treatment method that we use in Gulf War illness, an integrative medicine, sort of holistic and also layered approach to treating complex illnesses, they have embraced Gulf War illness into the clinic and changed the focus of that clinic to incorporate more than just the post-viral illnesses. And so that's exciting. And no further introduction. I don't want to steal any more time. Elizabeth.
Oh, thanks, Nancy. Let's see here. Is that showing up okay?
Yep.
Okay.
So good.
All right. So I know we're behind on time, so I'm going to try and keep this very short to try and get us back on track. But I'm going to summarize some of the preliminary results from a CoQ10 randomized double-blinded placebo-controlled phase 3 trial that has concluded, but the results have not yet been finalized. And so just to go over the background of why this was being looked at, and I know Dr. Colomb also mentioned they have an ongoing trial as well, and she has some of the preliminary background that I'll mention. But just briefly, we know that Gulf War veterans experienced environmental exposures that caused oxidative stress, some of which has just been reviewed. And one of the resulting mechanisms that's been seen is this mitochondrial dysfunction. And as a reminder, the mitochondria are the powerhouses of the cells. They're responsible for energy production. And so this feeds into issues of fatigue as well as all kinds of other system dysregulation when you have inappropriate mitochondrial function. And just one example of some objective evidence of this mitochondrial dysfunction was shown also by Dr. Golem's group, this Koslik article I reference, showing post-exercise phosphocreatine recovery time, dysfunctional in a Gulf War illness group. So how to go about treating this? CoQ10 is an essential component in the synthesis of ATP, which provides energy to cells and is involved in some of that mitochondrial function. So there's been a lot of interest in CoQ10 over the years in Gulf War illness, as well as in a number of other issues to try and improve some of this synthesis, and as well because of its antioxidant properties. And of course, it has an excellent safety profile. And is widely available and relatively inexpensive. Although, as Dr. Glow mentioned, getting high quality product may be more expensive. So in order to give a little more background, the main trial that's been done in Gulf War illness was done by Dr. Golem's group about 10 years ago, and that was a smaller phase 1/2 trial of 46 participants. And they did find when they did a stratified analysis, so when they removed the women, which was a small number, and looked at only the men, they did see improvement in general self-reported health overall in that CoQ10 receiving group. However, this was a small sample size, and it was clear that we needed a larger study to look into this some more. And I did just also want to note there was some thought of a possible correlation seen in the improvement in those symptoms with the serum CoQ10 levels, and particularly with a change from baseline to the treatment levels of CoQ10 that may be responsible. And then just very briefly to mention, that's there has not been a lot done in Gulf War illness, but there has been there have been a number of studies. This is not all inclusive. This is just a smattering of some that have been done over the last 10 years in ME/CFS and fibromyalgia, which of course are not exactly the same thing, but do share a lot of overlap. And in some of these studies, they've seen oxidative stress reduction and improvement in various symptoms, particularly in fatigue. I should note that both of those ME/CFS studies, however, were not done with CoQ10 alone. They were done one with in combination with NAD and another in combination with selenium. So this phase 3 trial set out to enroll 200 participants from 4 sites. It was a multi-center trial, Miami, Boston, Bronx, and Minneapolis. Unfortunately, recruitment was slow, and then because of the pandemic, recruitment was ultimately stopped early. So the final sample size is 100, which reduces the power of the statistical analysis we're going to be able to complete. But you can see that they were randomized to CoQ10 and placebo, and I'm just showing here a graph of the serum levels, so the blood levels of the CoQ10, and those in blue are the group that was receiving the CoQ10, and those in red are the placebo. And so you can see that the baseline ranges, the people's baseline level of CoQ10 ranges, but then when those are supplemented, you can see a clear divergence in the groups. Those who are supplementing are getting clearly increased levels of CoQ10. There was a run-in period of a higher dose up until 8 weeks, and then a dose reduction, which is why you see that peak and decrease. Unfortunately, the primary outcome, which is a change in baseline SF36 physical function score, which is a composite score of overall physical function and well-being that is done using a mixed methods analysis, sort of over time from that initial week 4 visit up until week 24, did not show a significant change. We did see a trend towards potentially improved scores in those who had the highest CoQ10 serum levels, although that is not a significant outcome either. And we're also looking into a number of other secondary and exploratory outcomes. So at the final time point, we see some potential improvement in natural kill cellular function in the CoQ10 treatment group when it's looked at both by randomization as well as when we stratify based on actual serum blood levels. We did not see any significant differences in other immune markers that were looked at, but we are currently exploring some other avenues in terms of the difference in that change from the baseline to those who have the lowest baseline CoQ10 versus treatment. And then, of course, this was a lot of work, a really rich data set. A lot of data was collected in terms of sleep and neuropsych outcomes. And we are looking into those both in terms of the effects of CoQ10, as well as just to continue to understand Gulf War illness overall and come up with additional pathophysiology and everything. So I'm going to stop sharing if I can find.
You did. You're good.
I did. Okay.
Sharing. Thank you, Elizabeth. That was great.
Thanks.
We're going to move right on into
Yep.
to Leo because we have time, and then we'll hopefully have a little Q&A at some point. So the next speaker is Dr. Leo Tamaras. He's MD with an MPH. He's a professor of clinical medicine and public health at the University of Miami. and he is in the leadership position at the Miami VA, is with the long COVID clinic and now this multi complex illness clinic with Elizabeth and with Dr. Palacio. So he's involved in much of the Gulf War illness research. I think that I haven't told you all that I was transitioning out of the VA after 41 years of service. And Dr. Palacio and Bast and Dr. Tamaris have picked up the Gulf War illness portfolio at the VA while I continue to have that portfolio at the university. So so anyway, I will let Leo take over from there.
Oh, thank you. So Dr. Klimas has done an impressive amount of Gulf War illness research for some time. And before that, she did MECFS work. So what we have been doing lately is trying to compare or trying to use the data that she's accumulated over time and try to compare different diseases, which are called complex medical diseases. One of those is Gulf War illness. And the first bit of results that I wanted to show you is what the function of natural killer cells is in Gulf War illness. What are natural killer cells? Natural killer cells should be known as your first line of defense. So if you are attacked by a virus or a toxin or something foreign in your body, the natural killer cell is the first cell that comes in and sees if that foreign part is actually foreign and if it needs any attack. So your natural killer cells would, if there's a foreign virus and your natural killer cell comes in and then identifies it as foreign, they would produce cytokines, which are inflammatory molecules, and they would call in the troops, and the troops would come in and do their thing and actually take care of the foreign virus or foreign bacteria or foreign toxic exposure. So natural killer cells are pretty important cells in complex diseases. And what we did is we took Dr. Klimas's data on natural killer cell function in Gulf War illness, in MECFS. We've done it also in long COVID and also in controls. And we measured how active those cells are, and that's what cytotoxicity means. So if your cytotoxicity of the natural killer cell is high, that's good. If the natural killer cell cytotoxicity is low, that's not good. That means that your first line of defense is low. And you can see from the table to your right that the cytotoxicity compared to controls, both in MECFS, which is chronic fatigue syndrome, or in Gulf War illness, that activity of the natural killer cells is low, which is not good, because that means that your first line of defense is not actually defending you well. And it's not only not defending you well, if you are not defending yourself well, you're not producing cytokines, or you're producing cytokines, too much cytokines, and that is going to impact how you defend from illnesses and how your body patrols your cells and identifies things that are foreign or not foreign. So as I was saying, so we're doing this and we're seeing that the same results in complex medical illnesses like MECFS, Gulf War illness, long COVID. So that means, what's the implication of this result? The implication is that your immune function in those diseases is actually affected. So now that I've said this, let me go to the next set of slides, which is about, and just bear with me, which is about a study, oops, my bad, which is about a study that was conducted using some medications that might improve the function. And this is the glutathione and curcumin trial in Gulf War illness, conducted by the principal investigator was Dr. Klimas. This was a multi-center study. It happened at 3 sites. And the premise of this study was that, as you know, Gulf War illness is an inflammatory disease. It is a disease that involves chronic systemic inflammation that affects multiple organs. And you can feel all this because of the symptoms that you have: fatigue, brain fog. So it's not only your muscles, it's also your brains, also your joints. So it is a multi-organ disease that presents with multiple symptoms, and as I said, fatigue, brain fog, pain, um, and so looking for anti-inflammatory strategies is probably a good idea. Um, and the 2 medications that were tested in this study were curcumin and glutathione. We know from prior work that curcumin in rats and in humans, it does have an anti-inflammatory effect. So it can curtail this inflammatory uh, um, part of Gulf War illness. It inhibits a family of inflammatory medications that's called NF-κB, and the goal, what curcumin can do is reduce pro-inflammatory cytokines like TNF alpha, interleukins 1 and 6. And therefore, by doing this, it can curtail some of the symptoms that can be produced by those high levels of inflammatory cytokines. Glutathione, besides having the same anti-inflammatory effect through the same molecule and family of molecules that activate the inflammatory system, it also has a role with vitamin B12 on DNA methylation. So making sure that your DNA is working well, and specifically in the mitochondria, as Dr. Bass was saying. So having a good mitochondrial function is probably pretty good, pretty important. And it does this, glutathione does this by making sure with vitamin B12 that there's DNA methylation in the mitochondria. So it not only can help with inflammation, but also might help with energy dysregulation. So this study was a phase 1 to 2 randomized clinical trial that compared 3 arms: curcumin, glutathione, and placebo, in veterans with Gulf War illness, trying to see not only clinical outcomes, but also trying to see if it impacted the mechanism of Gulf War illness. The main part of this, the main outcome of this study, or the main result of this study, is the dynamic modeling. What does that mean? The dynamic modeling is, for those of you who participated in this study, so you would start the study, and then we would put you on a bike, and while making you exercise, we will draw blood multiple times to see if exercise got better or got worse. And we would measure inflammatory molecules throughout the multiple times that we put you on the bike. The third outcome, or the third aim of this study, was to evaluate the methylation, and if it affected in any way the oxidative stress that we can see in patients with Gulf War illness. Study included about 90-something participants. These were young patients, 50 years old, who had no main comorbidities, had normal renal function. Their body mass index was around 30. The majority of these participants were male, and there was a good representation of minorities. The study, as I said, was divided into three groups: curcumin 30 people, glutathione 33, and then placebo 31. We did have loss to follow up in the study, which was not on one specific group. The loss to follow up occurred in all of the groups, and it was about 10 participants per group. The study did not show a change in symptoms. As Dr. Bass said, we had the same result. There was no improvement in symptoms after several weeks of taking the medications. There was a trend in increasing natural killer cell function. function. So it did improve, particularly curcumin. Both medications actually improved the exercise capacity and in the VO2 max, and we are pending the dynamic modeling, which is the main result of the study, and we'll come back to you and let you know what those results are, because those are particularly important, trying to see if during exercise your cells and your inflammation got better or worse with one medication, because that would tell you a lot about the mechanism of how the medications worked. And that's the end of my presentation.
That's fantastic. Thank you very much, Leo. And I think that it's one of the things it brings up when we're designing these studies is that we actually don't know what the best primary outcome variable should be. I mean, having someone have better exercise capacity may have been perhaps more important than some of the symptoms we were following. So that's one of the problems in a field where we don't have enough data to support what when we design our studies, what the best design might be. But I think it was really interesting in the patients in the study that they actually did have more exercise capacity. It's rather important. Our next talk, I'm going to move on because Q&As are at the end if we have any time, is going to move on to Dr. Christina Enley, Zach Barnes, and Lubov Nathanson. And I'm going to shorten these rips. I don't want to take all their time by introducing them. But I'm going to start with Zach because I've known Zach since he was a student, and he sure is not a student anymore. We go back way, way, way back. But he's been working with our group in many different capacities, including in our research team for 18 years. Dr. Ainley is our assistant professor at Nova Southeastern. She directs the clinical laboratory, and she has done a lot of work in her career on neuroendocrine disruptions due to many things, including toxic exposures and these complex illnesses. And Dr. Nathanson directs our genomics work here at the Institute for Neuroimmune Medicine. And she is an associate professor at Nova Southeastern. She's done a lot of work in Gulf War illness. I can't brag on her enough. She's done a lot of work on the genetics, the epigenetics, triggers, and we're just couldn't be prouder to have someone of her amazing scientific integrity as well as scientific skills to be working with our team. So I'll let them go ahead and start.
Can you hear me okay?
Yep.
Great. So my name is Zach Barnes. As Dr. Clemens mentioned, she's known me too long that she's thrilled to admit. And today I'll be talking about the Gulf War reboot study and how Gulf War illness in females is different than Gulf War in men. I'm lucky to be joined by Dr. Ainley and Dr. Nathanson, so I'll be introducing them as the talk proceeds. Today we'll be reviewing the progress of a second study of a multi-hit clinical trial in Gulf War in men, and as I mentioned, the differences in females with Gulf War illness. Now, Gulf War illness has clinical similarities to fibromyalgia and ME/CFS. So the key question becomes, how do you treat how do you move to treating the underlying mechanistic issues and not just the symptoms? And the way that is teased out is by examining the underlying biology. The way we do that in a controlled setting is by using an exercise challenge that has been mentioned several times during this conference. And as you can see, for example, perforin molecules per NK and CD8 positive cell, while looking similar at rest in Gulf War and healthy controls, starts to tell a very different story once you get into response to exercise, both at peak exercise and even lingering effects for hours afterwards. In this case, you can see that there is supposed to be a 50% increase as it is in healthy individuals, and then that comes, as a response to a stressor that then comes back down 4 hours later. However, in Gulf War illness, no such response is observed, and then there's actually a suppression 4 hours after. So exercise challenge allows us in a controlled setting to look at the underlying biology and the underlying symptoms that result both as a result of this key stressor, but not only that, but long-term after the fact. So as I mentioned before, the underlying biology it becomes pretty important, right? So what you end up seeing as a response to exercise specifically, you see that these different systems, what we call multi-symptom illness, the immune system, the neurologic, the cardiac pulmonary, the HPA and HPG axis, your ability to respond to stress is very different at both a intracellular level, which we saw with the perforin granzymes, the cell population level, what those immune cells, for instance, are doing, and then how they signal one another to act. As you can see here, healthy controls have a very different network than those of ME/CFS, which is a similar disease. And we, by using exercise, we collect this clinical and laboratory data before, during, and after to paint this picture using network analysis. Now, so the question becomes, okay, you study this, you see that there's differences. Well, how do you then model treatments? Because at the end of the day, that's what we're trying to do. Now, the way this occurs is you build what's called a system regulation model based on literature review. So anytime anything is published, you know, we in our team, the systems biology group, take that information and build connections between these different systems, the immune system, you know, the HPA, the HPG axis, and we try to see what those connections are, what's turning things up, what's turning things down, what's not changing, in order to assess how you can push people that are sick into a more healthy, steady state, a more healthy regulatory state. So what treatments could work? It's bottom line is it's complicated. Uh, you know, we use these models to give us the idea for a 1 and a 2 hit hypothesis, where you're taking not just one medication but a second medication in conjunction or in sequence to see how, and then without taking the blood of a single person, to see how that interaction changes in the model to see if we've moved towards a state of health. And with that, I will introduce our first speaker, who is Dr. Christina Ainley. Go ahead, Dr. Ainley.
Great, thank you, Zach. Can you hear me?
Yep.
Great. So I'm going to expand on this work and talk briefly about a wonderful study that we just completed at the Miami VA. This was our women versus men with GWI. Dr. Klimas was the PI. And Zach, I think you have control of the slide. Thank you. So Dr. Klimas and Dr. Fletcher over the years have a substantial amount of data for Gulf War illness, ME/CFS. And so some of the initial studies did find that we were beginning to see gender differences in GWI. Looking at baseline, so these are individuals at rest, we saw differences in cytokine. Now, as we take this data and we start introducing it to the modeling, you can see where we have to adapt between the male and females and potential treatment differences. Not only due to changes in hormone signaling, but as we build the model, we're finding more key differences. So the woman versus men study is another exercise challenge, so dynamic modeling, 9 time point. So the women come to the Miami VA at T0, that's baseline. That's our initial blood draw. T1 is VO2 max. They're on the bicycle, and T1 is when they hit their peak performance. And then we collect the blood at various time points, including 1 hour post T1, 4 hours, and they come back the next morning where we get 24 hours after VO2 max. So this is the goal, this complicated hodgepodge of networking. And we've done this across our studies, but what we really want is to build a model specifically for the Gulf War women. So we're looking at hormones, we're looking at cytokines, signaling factors, cellular response at all 9 time points. It's a lot of data. We have it, we're building it. I don't have time to go over all of it today, but I do want to focus on a few key points. In particular, one of the cellular responses, which Leo just beautifully introduced, is NK cell function. So as Leo said, these are cytotoxic lymphocytes. They're the ones who rapidly respond to a viral stress, stress cells, pathogens. We use a chromium 51 assay. Okay, so we're looking at NK cell function. In addition to this, we also look at the number of NK cells that are circulating in the blood. And what we have found across our subjects, across studies, is that the number of NK cells that are circulating in your blood are normal. You have a normal number of cells, but in Gulf War illness, they're not working as well, similar to what you see in ME/CFS. So next slide. Here's our data from women with Gulf War illness. And you can see at T0 at baseline, they have a significant lower level of NK percent, that's function. At baseline, their cells are not working as well as the healthy control women. Now we go across the exercise, and sorry, perfect. As we go across the exercise, that continues to stay lower, and we see a significant drop at 1 hour post-exercise, and then a slight back up at T3. So our Gulf War females, they come into the exercise challenge, which is very challenging. This is a difficult task. They come into it already with a lower, with their NK cells at a disadvantage. Now, trying to figure out why this could be, what's happening. Next slide. We look at cytokines. So one of the cytokines that we saw a significant decrease in our Gulf War women was IL-10. So at baseline, the level of IL-10 is decreased. And then we see a decreased level at T1 plus 60. Okay, so 1 hour post, it's also different from the controls. Now, IL-10 is very interesting because IL-10 can induce NK cells, and NK cells can produce IL-10. Next slide. So coming back to our complicated and very extensive network, we're adding in all of this data: hormones, cytokines, cellular response, and building the picture, building the modeling. Now, we are putting a particular focus on the hormones, right? As our Gulf War women are aging and they're transitioning from perimenopause to menopause, postmenopause, so we need to adapt the modeling and the treatment paradigm. So all of that is coming into the modeling to really help fine-tune potential treatments. Now I'm going to leave it here. Thank you very much, and thank you to all of our Gulf War female veterans who participated in this study. And next up is Dr. Nathanson to go over her amazing genomic work.
Good afternoon. If it is possible, I would like to share my screen.
It's shared. You're doing well.
No.
No, it's not. Yeah, take your screen down.
Thank
Oh,
you.
there you go. Thank you, Zach. Yep, now we see your email.
Sorry.
Try again.
1 second.
It's hard to get the right window when there are thumbnails.
Yeah, okay. Yes, now you should see my slides. Okay,
Yes.
so my part, we for genomic, for transcriptomic and epigenetic part, we used a subset of data of samples. And in our study, we used samples from 25 women with Gulf War illness and 18 controls. And we had these participants. They are a subset of these larger studies. And as you can see, all participants had about the same age and BMI, but females specifically were different. Females with Gulf War illness diagnoses were different from their healthy controls, even more different than males. Okay, so you already heard enough about this exercise challenge. I just wanted to say that for transcriptomics and epigenetic studies. We used baseline T0, we used T1 time point. It is maximal exertion, peak of exercise, and 4 hours after peak of exercise. So from each blood sample, we selected PBMCs, and from PBMCs, we isolated RNA and DNA. So DNA from baseline, we studied DNA methylation. We already published our DNA methylation data in males with Gulf War illness, and comparing females and males is now under analysis. But today I will talk about RNA sequencing and microRNA. So microRNAs, it is very important part of epigenetic regulation. So RNA sequencing would show us which genes are fired up, which genes are silent, and microRNAs, they will show what is upstream regulation of all these processes. Okay, so we compared differences in females and males in healthy controls and in Gulf War illness patients. And I will not talk about specific genes, but I want to show you really pathways, metabolic pathways, because actually this what matters. So here it is difference between these two time points, between baseline and peak of exercise. So here I want you to pay attention to this pathway. So if it is really dark, so these pathways were really very much affected. If it is very light, it was not affected, it was not changed. And as you can see, in Gulf War patients, difference between males and females for many pathways is absolutely opposite. And for many immune pathways, healthy males and females have about the same changes, but for Gulf War patients in females, mediate immunity, lymphocyte immunity, leukocyte immunity, and interleukin 12, it is not responsive to peak of exercise. So one of our explanation was that females with Gulf War, they have very strong fatigue right from the beginning, and they already had all these pathway changed right on baseline. And I wanted you to pay attention to this difference in interleukin 12. Interleukin 12 actually stimulates proliferation and differentiation of T cells and NK cells. It has a lot of different immune functions. So as you can see in healthy males and females, by peak of exercise from baseline, it is a lot of changes. In females with Gulf War illness, almost no change. Now, we used this data, RNA sequencing data, and we looked at cells. So this analysis, it was not done by flow cytometry. It is derivative of RNA sequencing data. And here, as you can see, differentiation and proliferation of CD4 T cells is different in female patients with Gulf War illness, and probably it is because of changes in pathway of interleukin 12, probably. So in healthy females and males, changes are about the same. So in Gulf War illness patients, it is different in females and in males. Okay, so now we are moving to differences between peak of exercise and recovery. It is 4 hours after peak of exercise. So basically it is recovery. And here I also want you to pay attention at some pathways that change very differently in females with Gulf War, and how different these changes are from changes in healthy controls. So again, there are a lot of immune pathways that there is no change in females with Gulf War illness. And in healthy females, normal change upon recovery. Oxidative stress induced senescence. So well, this is normal difference between males and females, but as you can see, females with Gulf War illness react to recovery very differently, and there is a lot of changes in oxidative stress response in females with Gulf War illness. Okay, so these results we also used to look at cells, and we had talks about changes in different immune cells. So here, what you can see in females with Gulf War, decrease in dendritic cells upon recovery, and this decrease is very statistically significant. So there is some decrease in healthy controls, small decrease. There is also decrease in males, but it is not really statistically significant. In females with Gulf War illness, decrease in dendritic cells is very significant, and it is statistically significant. And probably it is due to dysregulation of estrogen. Because actually estrogen normally does regulate development of dendritic cells and proliferation and development of eosinophils. Okay, now to epigenetic part, microRNAs. So microRNAs, those are small, very small RNAs, and their variation in evolution. They play a lot of roles in whole metabolism in our cells. And they regulate many different processes. They regulate methylation of DNA, they regulate synthesis of proteins. So synthesis of proteins is probably the best studied function of microRNAs. And here I want to show you changes in 4 microRNAs. So it is microRNA 188, 140, 152, and 136. So we'll start with microRNA 152. Actually, this microRNA, it regulates epigenetics. It can regulate DNA methylation, and it also regulates inflammasomes. So it can suppress inflammasome activation. As you can see, at all 3 time points, this microRNA is decreased in females with Gulf War. So in control females, it is at higher levels at all 3 time points, and absolutely different picture in males with Gulf War illness and with healthy controls, almost no changes. Okay, these 3 microRNAs, 188, 140, and 136, these microRNAs, they participate in a lot of immune functions. For example, 136, it regulates NF-kappa B signaling and production of cytokines. This microRNA 140, it can suppress NF-kappa B signaling. It is anti-inflammatory protective. And this one, it actually affects biology of CD4 T cells. And you can see that at all time points, this microRNA, it is higher in females with Gulf War illness than in female controls. And in males, this picture is absolutely different. In these two microRNAs, 140 and 136, picture is opposite. So these microRNAs, they are lower in females with Gulf War illness than in controls. So this
Yuval,
is,
I'm going to speed you along a little bit.
yeah, I'm finishing. So this is a lot of data. And basically, what I want to say in the end, when it comes to diagnostics and when it comes to treatment, it is very different in females and males with Gulf War illness. So it is on molecular level, on the level of nucleic acids, as well as protein levels. Okay, so I stop sharing.
So if you were to summarize what you just said for men versus women, what would you say the take-home point was?
Take-home point is females and males are very, very different, and we have to study more. So this can be considered as only pilot projects. There are a lot of questions why it happens. So we have to repeat these studies on much bigger cohorts and look at everything else.
Great, thank you. Okay, Zach, I think you wanted to wind the session, this particular set of talks, not the session, up because Dr. Chang still needs to talk. But we're short on time, so keep it quick. Is that your slide, Zach? Systems biology? Because we don't hear you. You're muted, Zach. No one can hear you. No, you're not muted. Can you hear him?
I can't hear him. No, it says that he's muted, but I see his screen.
Yeah.
Is this better?
Yes, thank
Yeah,
you.
we can hear you now.
Sorry about that. All
you're
right,
good.
so basically, what we're looking at is a complex system. You can think of it like an engine, right? And in the case of Gulf War, the engine's simply not running the way it should be, and you're stuck there. So the way that we visualize this is imagine peaks and valleys, right? And you're stuck in these valleys. Each of these valleys here in figure A is a valley of what you consider to be a sick state, right? So no matter what you do to nudge yourself out of this state with whatever medication, you end up in the same spot, right? So the idea of what we're trying to do here in this first integrated modeling study and then the subsequent reboot study is to model a path out of the valley, right? Out of these theoretical sick states. In order to do that, what we need to do is we need to push you over the mountain and keep you in the valley that we want to keep you in, and not get you stuck in a different state, right? Avoiding these other illness states. So the way we do that is with the integrated modeling, right? A 1-month intervention of TNF antagonists, which at first look had small improvements, but not what we were looking for. So we decided to take a closer look and learned that actually, as we predicted, there were those that responded and those that did not respond. The responders exhibited between a 10 and 30% increase in vitality, which is actually exciting. So we decided to take an even closer look at these individuals. Found that things like emotional limitation and well-being scores were improved. There was reduced physical fatigue, and the reduced activity metric was lowered as well. So what else happened with these responders? We looked more closely at them, and we saw that we were nudged in the right direction of a healthy state, but not enough, right? So we went back and we said, what can we do? We went back to the model, the simple model, and we said, let's add elements, let's add information that gave us more clarity. It gave us more defined mechanisms of information exchange and better feedback and predictions of these steady states. And what that told us is that as we moved people in this protocol, they took their their month of treatment, we nudged them so far out of the valley that they were actually into this other space after 6 weeks. And then at the end, this is again 6 weeks after not taking any treatment, they were actually moved 20% closer to a healthy state, which was better, but again, not enough, right? So that's why the reboot study happened, which was we increased the dosage and the time in the new clinical trial that the intervention occurred. Again, we viewed responders, right? This time at a better clip, we saw that there's actually a 10 to 40% improvement in those that responded, right? The network analysis is underway, but we need to get the timing and the dosage correct. So, and therefore there's another analysis that needs to occur, which is what we're doing now, right? More info isn't enough, right? We need to change how we view the information that's exchanged. And that's what David Casada in the systems biology group at the INM at Nova Southeastern, he's not just adding the direction that these cell populations and these cytokine signals direct, like positive or negative, but he's looking at the size of the effect and how long the duration of those effects, right? And that allows for the accurate recommendation of dosage and treatment lengths when we're designing these clinical trials, right? And so whenever you get these connections with direction and magnitude, timing and dosage becomes a real possibility at a much larger scale, which is going to at the end of the day, what's the takeaway? It's going to fast track treatments. It's going to fast track results. That's the end of our presentation.
Thank you.
I'd just like to thank everybody, specifically the participants in these studies. Without the participants that are getting on these bikes, being exhausted for weeks at a time, being poked and prodded, without this, we can't get the treatments into your fellow veterans' hands. So thank you, everyone.
And such an important thank you. I appreciate that. And then there's another take home that after you saw that computational modeling, that was a successful study that we predicted a 40% response rate and we got 60%, which is better. And so we are going to be moving that to phase 2 if we can successfully compete for funding. So that's exciting study. And thanks to all the people that participated in the first and then the second phase of that study, we'll be able to move it on without the bike. So that'll be easier on people, but it is this reboot study is one of the more exciting studies that we've been involved with, and we couldn't be more thrilled. So that was a whole host of things. We still have one more talk in this session. I know we're running over by quite a bit, but not horribly. We're going to punt our Q&A question to the chat. If people have questions, please put them in the chat for the investigators and the speakers, so that we can begin to catch up again. The last speaker in this session is John Chang. John Chang is a fourth-year med student at Nova Southeastern University. He's doing a gap year, doing a whole year of research with Dr. Galore, who is a professor at the University of Miami and works with the Bascom Palmer at the Miami VA and Bascom Palmer Institute. And she's done some exciting work in Gulf War illness, perceiving the eye as a window to the brain, and doing work that used her expertise as a